What do you think of the format? You like it, not like it? Like format? No. Hate it. Okay, what did you not like about it? You didn't like the matching. Terrible what? Well, they all had a match. Okay. So you would like me to eliminate the matching next time? Why don't I just give everybody 30 points? Okay. I'm not tied to that. We don't have to do matching. No, we can't do essays. I don't have enough tea. It'd be a month before you got them back. I can do more short answer.
It was a lot harder because it was so much to read through before you could make sure that you're picking the right one for every single question.
For the matching you're talking about. Okay.
Yeah, it's easier one letter.
Okay. Let me think about it. I guess my gut reaction is I just make the fill in the blanks more of those would be my inclination. Let me think about it. If you want to send me suggestions, I'll be happy to take a look at those. How's that? Yeah, but when you do that, so what she said was if I had like 8 choices for 5 answers, when I do that, then what happens is the guessers start benefiting from that because it's more likely they're going to get it. And so I want the matching or whatever it is to benefit people who know, not the people who are guessing. So that's why it's got the setup that it does. We don't have to do matching, but I don't want to have only a very small number for there because I don't want to benefit the guessers. Yes? Well, those, okay, so the first part is, they're not multiple choice. The first part is really true-false. Each one is a true-false. You circle if it's true, and you don't circle if it's false. I'm really not inclined to have more than 20 of those, because that means that the guessers are going to get 10 points. That's what it means. And so if I go 40 of those, the guessers are going to get 20 points. And so I think the 20 is just about the right number of those. So you're going to guess half of them right is what I'm saying. Yeah. Okay, well, thanks for the feedback. That's the first time I've ever had a class that didn't want to do that, so that's interesting. Jessica?
I really benefit from the diagrams. I don't know if there's a way to incorporate that.
Diagrams?
I don't know if
Yeah.
it's too much or...
Diagrams are tough to do because they take up space, and if I put a figure in for one question, then if I do that for several questions, all of a sudden I'm adding several pages of stuff, and I'm just a little hesitant to do that, to be honest. I mean, in some cases, I think a diagram is appropriate to ask a question for, and I won't rule that out, but I can't do a lot of them. Okay, well, thanks again for the feedback. You guys have been a very interesting and a very good class in this respect. I really appreciate that, and I will give some thought about it, and before the next exam, I'll let you know where I'm at. It won't be a surprise. How's that? Well, other than the material. Okay, the exams are not graded, unfortunately. The TAs have been busy. I told them I need to have them by Monday, and so they are working on those, and I think they should be available by, not think, they definitely should be available by Monday. I'll put a note out confirming that on email, and you'll be able to pick them up in the BD office, but please don't go there until you get that note from me, okay? I will post a key outside my office when those become available as well. Okay. Well, fatty acid metabolism. So we're still in metabolism, and we're going to be in metabolism for a little while yet. We have fatty acid metabolism to do, and we also have nucleotide metabolism to do and prostaglandin metabolism to do. So we've got a little ways to go in metabolism before we finish metabolism for the year. Fatty acid metabolism is important, but I want you to keep in mind we're talking about fatty acids. Okay? So people frequently blur the lines in their heads between fatty acids and fats. Fats, of course, are compounds that contain fatty acids and glycerol. And fatty acids are the sort of meat component of the fat, but nonetheless, they are different things. So a fatty acid and a fat is not the same thing. Fats are important, obviously, for energy storage. Fats are problematic in our body because, again, fat is not water soluble, but the body needs to be able to handle it in some way. One of the ways in which the body handles fat is in specialized cells called adipocytes. A-D-I-P-O-C-Y-T-E-S. Adipocytes are specialized cells that hold fat. You can see an adipocyte right there, okay? And you can see that glob of fat right there. The adipocytes are holding that fat until the body needs the fat. And as we shall see, release of fatty acids from that fat in the adipocyte occurs as a result of action of lipases, one of which is hormone sensitive. And we'll talk about that briefly, not much depth. The thing to keep in mind about fatty acids is that fatty acids, like fats, also are a problem in the body, but for a different reason. So fat, of course, is a problem in the body because it's not water soluble. Fatty acids are water soluble, or at least they can be water soluble. And the problem with fatty acids is that fatty acids are what we use to make detergents. And detergents, of course, will denature proteins. So the body is not only careful with how it moves fat, because it has to be, it's also careful with how it moves fatty acids. And interestingly, we have a protein that's very abundant in our bloodstream, that one of its primary functions is actually carrying fatty acids. That protein is known as serum albumin. I'm sure you've heard of it before. And serum albumin carries free fatty acids in the bloodstream. Okay? So that by doing that, you're avoiding the accumulation and detergent effects of the fatty acids, and the fatty acids get to their target tissues as a result. Okay. Now, here's what we're going to be talking about today and on Monday. So, if we look at fatty acid oxidation and we look at fatty acid synthesis, what we discover is the two processes, at least from a chemical perspective, are almost identical, just the reverse of each other. Okay? Almost identical, but the reverse of each other. So, here on the left, we see fatty acid oxidation. The process of fatty acid oxidation, at least the main process of fatty acid oxidation, is known as beta oxidation. And it's a process that occurs in chunks of 2 carbons at a time. Okay, so here's a fatty acid that goes through an oxidation reaction. The oxidation reaction occurring here with this formation of this double bond. Hydration to put a hydroxyl on there. Oxidation to get it, and I'll talk about these in detail so you can just kind of follow me for the moment. Oxidation to get to a ketone, and then cleavage to generate an acetyl-CoA. So fatty acid oxidation is leading to acetyl-CoA, and an acyl group that has 2 fewer carbon atoms. Okay, so oxidation leading to the formation of acetyl-CoA. When we look at the synthesis of fatty acids, we start slightly differently in that we start with a 3-carbon molecule, as we will see, but only 2 of those carbons actually get put on. So 2 of the carbons get put on, and as a consequence, we see essentially the reverse of the previous reaction with 2 carbons being added. We've now got this ketone again that we had before. Instead of doing oxidation going down, we do reduction going up. Instead of doing hydration going down, we do dehydration going up. And finally, we do reduction, which is the opposite of oxidation, going up in the direction of fatty acid synthesis. Now, there are other differences, but from a chemical perspective, they're essentially the reverse of each other. The other differences relate to some of the cofactors that are used and so forth, and I'll talk about those. But from a chemical perspective, the two processes are essentially the reverse of each other. Okay. Now, interestingly, there's little regulation that's involved. There's one enzyme in fatty acid synthesis that is regulated. It turns out to be a pretty important one. And there's only there's nothing in fatty acid oxidation that is regulated. Once you have free fatty acids, they will be oxidized. So the only regulation in terms of breakdown of fat all the way down to acetyl-CoA, the only regulation that happens is at the regulation of the lipase that breaks the fat down. And I'll show you that in a bit. So that once we've got fatty acid, no regulation at all. It just gets oxidized. Okay. So that's a sort of a quick and dirty overview of what's there. Fatty acids, not surprisingly, tend to have even numbers of carbon atoms. Think about it. We're adding 2 at a time. Okay, you can do the math. We're going to end up with an even number of carbon atoms. Most of the time, it's not 100%, but most of the time we have even numbers of carbon atoms. And we break them down 2 at a time. And everything works out. Okay? There's a famous experiment that a man named Knoop, K-N-O-O-P, did around the turn of the 20th century, where he basically demonstrated this 2-carbon phenomenon. And he took, and it was one of the first experiments where they used a sort of a biological labeling of a molecule to trace what happened to it. And he actually added molecules that had benzenes on the end of fatty acids, and he fed them to dogs and looked to see what he got out. So if he fed it with an odd number of carbons attached to the benzene, he had 1 carbon that was left at the end, what the dog excreted in its urine. And if he had an even number, it had 2 left on the end. So he knew from that these units were taken off 2 at a time. It didn't matter what the length was. It only mattered if it was odd or even. So, and of course, we know this now today from a variety of other experiments, but an interesting experiment that's over 100 years old, or about 100 years old. Okay, well, in the last lecture, I talked about the importance of the bile salts, the bile acids, which are essentially the same thing, in solubilizing fat as the fat enters your system from your diet. Glycocolate being one of those molecules, and I trust you remember that comes from cholesterol. And that coming from cholesterol with some addition of different groups makes this a fairly polar molecule. And this fairly polar molecule acts as a detergent to help solubilize fat in the movement through the digestive system. As I mentioned last time also, fats take an unusual path in our body from our diet. We eat them, they enter our digestive system, and there are lipases that take off a couple of the fatty acids. Then everything is moved across the intestinal wall, and then these guys get attached back on to sort of recreate that fat. And those reactions that I described last time are actually what you see on here. So these reactions on the screen are occurring in the digestive system. There are other reactions that will occur when the fats get to their target tissue. Okay? This, again, schematically showing you the last thing I just described. So here's the triacylglycerol. Here are the lipases. This is in the intestines. Okay? We see the movement now of the fatty acids, the movement of the monoacylglycerols that's left, the resynthesis of the fats, and now the packaging up of these guys to the chylomicrons, and ultimately to the lymph system, and then finally to the blood system. Yeah?
So it breaks off 2 of the fatty acids, but the glycerol, when it moves across the membrane, it still has 1.
Yeah, so it removes 2 of the fatty acids, 1 remains stuck on there, Carl, that's right. Okay? And they all get put back together to make the original fat once they get across. Okay, well, here's our friend. You've seen something like this before. You thought, well, I've seen the last of that last term. I'll never have to look at that again. But now you can thank your lucky stars that, wow, I memorized that last term. I'm not going to have to memorize it this term because there's some very similar things going on. You saw last term how we had 7-TM receptors and 7-TM receptors that bound to hormones. We talked last term about either glucagon or epinephrine that can stimulate this process. And that's actually the process that's going on right here as well. Okay, there are other hormones and other receptors that can also be involved. What you're seeing is a pathway that is activating an enzyme in a target cell. Okay? This target cell would have this receptor, and most commonly it would be an adipocyte. Okay? So here's the target cell. Binding of the hormone stimulates activation of a G protein. G protein activates adenylate cyclase. Adenylate cyclase makes cyclic AMP. Cyclic AMP, in turn, activates protein kinase A. And protein kinase A puts phosphate onto triacylglycerol lipase. Okay? That addition of the phosphate causes it to be activated, just like we saw the glycogen phosphorylase being activated. And what this enzyme does is it clips one fatty acid off of that fat. So this enzyme right here is the only enzyme that is regulated in the breakdown of fats or fatty acids, triacylglycerol lipase. You sometimes see it referred to as hormone sensitive. In fact, that's probably a better name for it. Hormone sensitive triacylglycerol lipase. The hormone, of course, activating it here, and we see this occurring all the way through here. Now, once we've gotten that first fatty acid off, we're left with a diacylglycerol, and then other lipases can cleave it all away. Okay? So once we've got the first fatty acid off, other lipases, which are not regulated, can then break the rest of it down. Yes, Lori?
Does it matter which fatty acid you take out first?
Yeah, actually, you will see the one that you would consider on number one, the top one. So that it would always tackle the one on the end. From the perspective of the enzyme, though, one end is equal to the other, but it won't get the middle one.
So it'll either get essentially one or
Yeah, but what I'm saying is the enzyme can't tell what is 1 and what is 3. It's the same thing. So it'll get an end one, but it won't get the middle one. Yes, sir?
You said this is going on in
Typically in adipocytes, yeah. Typically in adipocytes. So now the adipocytes have gotten a signal, we need energy. We're going to break down this fat, and we're going to release these fatty acids so that they can go out into the bloodstream and go feed cells. Yes? What kind of hormone? It could be. It could be an epinephrine. It could be a glucagon. It could be other hormones that we haven't talked about. But those are all possible for controlling this. Yeah. So one of the reasons losing weight, I should say, which is also losing fat, is slower and more difficult, is you actually have to activate this process in order to start the breakdown. So if you don't activate this process, then you're not going to use that. And it kind of makes sense to do it. The fat has a lot of energy in it, and you don't want to just willy-nilly start breaking down fat when you don't need to be doing that. So this process has to be activated in order for the fat breakdown to occur. Okay. So that's what's happening in the control. And as I said, the triacylglycerol lipase there is the only regulated enzyme in the fatty acid or fat breakdown. This is the reaction that, or actually the set of reactions. It's kind of a dumb figure, but it's just illustrating to you that there's the fat that we have on top. There's the action of 3 different lipases. It's not one lipase as it shows here, but 3 different lipases, which are in fact now leaving you with glycerol and 3 fatty acids. Now, if you think about this, if you look at the byproducts of the breakdown of the fat, these guys, when they're oxidized, are going to produce nothing but acetyl-CoA. We're only going to get acetyl-CoA out of these guys. Okay? This guy right here is the only part of a fat that can be converted into glucose, in animals at least. Plants and bacteria, they've got the glyoxylate cycle. They can use acetyl-CoA to make glucose. Okay? We can't do that. So the only part that we can use is this. This can be converted into glyceraldehyde 3-phosphate which is used in gluconeogenesis and can be converted into glucose. So only a tiny portion of the fat molecule can be used for immediate energy, the glycerol. That's the only part that can be used for that immediate energy that's needed by the body. Okay, and the mechanism by which that is converted is shown here. Here's glycerol. We add a phosphate to it, make glycerol 3-phosphate. You've seen this guy before. Okay, glycerol 3-phosphate can be oxidized to either DHAP and or glyceraldehyde 3-phosphate. And of course, both of these are intermediates in the gluconeogenesis process and can be converted ultimately into glucose if that's what the cell chooses to do. Okay, moving right along. This shows, I think, a broad overview of these processes. So keep in mind, here's our adipocyte over here. We've broken down. We've sent to various tissues. The glycerol is going to go to the liver. Anybody know why it's going to go to the liver? That's where gluconeogenesis is going to be available. Okay, so this glycerol isn't going to be of much value to the other cells because they don't typically run gluconeogenesis. All right? Whereas the fatty acids are going to other tissues because they are, in fact, energy sources for those other tissues as needed. Acetyl-CoA can be oxidized in the citric acid cycle. I can't pull it out. Okay, well, let's think about what happens during oxidation. So I've already told you that cells don't want to have free fatty acids floating around very much. All right? Outside of the cell, in the bloodstream, we package them up with serum albumin. Inside of a cell, okay, once they've gotten out of the bloodstream and into a cell, we also don't want them accumulating because we don't want those fatty acids denaturing proteins. That would not be a good thing to do. And so what cells do is they put a handle onto those fatty acids, and we see our friend coenzyme A, or CoA, entering the picture again. CoA, when attached to the fatty acid, makes what's called an acyl-CoA, ACYL. And the ACYL portion refers only to the fact that this is a fatty acid instead of an acetate. So acetyl-CoA means we've got acetate. Acyl means we've got a longer fatty acid out there. It could be propionate, or it could be palmitate, or linolate, or something like that. Now, in fatty acid oxidation, all the oxidation occurs on acyl-CoA groups. All of the oxidation occurs on acyl-CoA groups. And fatty acid oxidation also occurs in the mitochondrial matrix, which is really convenient if you think about it, because the mitochondrial matrix is where the citric acid cycle is going on. And the citric acid cycle needs to be fed by acetyl-CoA. And, of course, acetyl-CoA is the byproduct of fatty acid oxidation. So that's all very good. One of the unusual aspects about fatty acid oxidation, though, is how the fatty acid actually gets into the mitochondrion. So fatty acids get transported across cellular membranes. We won't talk about the mechanism by which that occurs. But they get into the cytoplasm of a cell, and the first thing that happens is that the cell will take that fatty acid like we saw in the last figure and put a CoA group on it. So when the fatty acid is floating around the cytoplasm, it's got a coenzyme A attached to it. However, to get across the mitochondrial inner membrane, that CoA has to be taken off. There's not a transporter that will move that guy across with a CoA on it. So the cell went to all that trouble to put a CoA on when it got in, and now when it gets to the mitochondrion, it takes the CoA off. And it replaces it with another molecule called carnitine. Okay? So carnitine takes the acyl group and attaches it. So we have an acyl carnitine at that point. And that acyl carnitine can, in fact, be transported across the inner membrane via a transport protein called a translocase. Once it gets into the matrix, the reverse thing happens. So the reverse thing in this case now takes the acyl group, attaches it to a CoA, so we end up with an acyl-CoA right here. That also generates free carnitine, which gets kicked back out into the cytoplasm. Okay? So this carnitine is functioning simply to carry the acyl group in. You can think of these two enzymes, if you want to, as essentially the same enzyme, just running the same reaction either forwards or backwards, depending upon your perspective. Okay? So pretty straightforward what's happening here. The net result is now we've got an acyl-CoA that's in the mitochondrion that now can be oxidized. Okay, so let's take a little closer look at that. That's not very good, is it? A little closer look at that oxidation, and we'll do it step by step. So let's take a look at what happens inside of the mitochondrial matrix. There are 4 enzymes in that matrix, and there's only 2 of the enzymes that I care that you even bother with the name about. The first is an enzyme that is important. Its name is important. It's called an acyl-CoA dehydrogenase. Excuse me. An acyl-CoA dehydrogenase. It's catalyzing this reaction. And no, I don't care if you memorize this name. All right? That's not important. The general structure, yes. Okay? The general structure, yes, is important, and I'll show you what's the important part of it. When we're looking at fatty acid oxidation and we're looking at fatty acid synthesis, all of the action occurs between carbons 2 and 3. That's where you want to focus your attention. In both fatty acid oxidation and fatty acid synthesis, the action occurs between carbons 2 and 3. Okay. If we look at this as the alpha carbon, we refer to this as the beta carbon. And this process is known as beta oxidation. That's where it gets its name. It's called beta oxidation because the oxidation is occurring around this carbon. Okay? All right. I like to think of it as 2 and 3. All right. Well, actually, technically, this is the alpha carbon, this is the beta. But suffice it to say that the name is deriving from the fact that the beta carbon is involved in the oxidation. It's actually, this is the beta here. All right. Now, the important thing is where the oxidation is occurring. So the oxidation has taken 2 electrons and 2 protons from here and has put them onto FADH2. Back when we talked about the citric acid cycle, I pointed out that the oxidation of succinate, catalyzed by succinyl-CoA dehydrogenase, had an almost parallel reaction in fatty acid oxidation. This is the parallel reaction. Because if you remember your citric acid cycle, succinyl-CoA dehydrogenase uses FAD. It makes a trans double bond at positions 2 and 3 of succinate. So exactly the same sort of thing is happening there as is what is happening here. The product being FADH2 and an unsaturated fatty acid. Notice that the bond there is trans. I'll talk later about trans fats and trans fatty acids, but when you hear about trans fats and trans fatty acids in diets, this is not the source of them. This is a very natural sort of structure. Okay? Has nothing to do with trans fats. 0. Trans fats arise from chemical treatment of food, and I'll talk about those later. Okay, well, there's this trans bond. Now, this enzyme is interesting. Okay, it's interesting because it turns out that there are 3 forms of this enzyme. They're all known as acyl-CoA dehydrogenases. The 3 forms work preferentially on long chain, meaning anywhere from about 14 up to, or maybe about 16 up to about 26 carbons. Medium chain that work from about 16 down to 6 or 8 carbons, and short chain that work on anything less than about 6 to 8 carbons. And you notice I had said general ranges there. So the acyl-CoA dehydrogenases work on short, medium, or long chain fatty acids. Okay? It turns out that long chain fatty acids, longer than 16, are not oxidized in the mitochondrion. They're oxidized in a different cellular structure, the lysosome. So oxidation inside of, I'm sorry, not lysosome, peroxisomes. Sorry. So the oxidation of longer chain fatty acids is occurring inside of peroxisomes. And the reason it's occurring there is that's where the long acyl-CoA dehydrogenase enzyme is found. So when these guys get to be oxidized, and I'll show you that later, when these guys get to be oxidized, they're moved to the peroxisomes first. The long ones, that is. The medium and the short chain acyl-CoA dehydrogenases are found in the mitochondrion. Now, one of the interesting things about these enzymes is if we look at the medium chain acyl-CoA dehydrogenases, this deficiency or shortage of this enzyme has been implicated in sudden infant death syndrome. Okay? It's not a cause-effect, but there are many cases where there is a relationship between either low levels of the medium length or no levels of the medium length and sudden infant death syndrome. So, sudden infant death syndrome can, in some cases, be linked, then, therefore, to fatty acid oxidation. Of course, fat metabolism and fatty acid oxidation. Okay. Questions on that? Yeah, Jenny.
Why are the long chains done in a different place?
Why are they done in a different place? It turns out it's not apparent up front why, but I have to talk about that when I talk about synthesis. And let me answer that question when we talk about synthesis. It turns out, and I'll tell you the answer, I guess, briefly, is that longer chain fatty acids are actually made in the mitochondria, which also seems like this. So then the question is, well, why are they made in the mitochondria? And I can't give you that answer. But longer than 16, one of the things that's made in the mitochondria is that. And so it's a very odd arrangement of these things. So it makes sense then to keep the oxidation enzymes for the long ones sequestered elsewhere. And that's what they do. Okay. All right, there's the first reaction of fatty acid oxidation. Second reaction. Very simple. We add water across the double bond. When we add water across the double bond, the hydroxyl goes on to carbon number 3. The hydrogen goes on to carbon number 2. We now have single bonds, and we have a hydroxyl there. And again, the name is not important. It's the general structure that I'm interested in. The general structure, you'll notice, this is an asymmetric carbon. 4 different things on this carbon. We have to give it stereoisomeric specificity. It is the L configuration. The L is the configuration of that hydroxyl on carbon 3 in fatty acid oxidation. We'll see that in synthesis, it's the D. That's one way you can tell synthesis from oxidation. So we have the L form of the hydroxyl on position 3 in fatty acid oxidation. Well, that hydroxyl group is, of course, a target for oxidation itself, and we see that happening in the next step. Now, let me back up. I meant to point out something here. Sorry. In that last reaction, we're basically following the citric acid cycle. The citric acid cycle, remember, we made succinate, which had a trans-double bond, and we added water across it to make fumarate. I'm sorry. Back up. I said that wrong. So we made the double bond. That was fumarate. We add water across fumarate. We get malate. Okay? It's exactly the same thing that's happening here. We're adding water across the double bond. We're making the equivalent of malate, but of course we've got a long chain out here. But the same sort of general reaction is occurring. And if you think back to your fatty acid, to your citric acid cycle, you recall that we oxidize malate to oxaloacetate. That actually makes a double bond back there, double bonded oxygen in oxaloacetate. And the same thing has happened here. Okay? The enzyme here is not important. Enzyme name is not important, as it was not in the last reaction either. The addition of the water, the enzyme name didn't really matter. But this is a second oxidation. NAD is going to NADH, just like it is in malate going to oxaloacetate. Okay? Now, we're almost done with the oxidation pathway. The next step will involve what's called thiolytic cleavage of the bond. And unfortunately, that's the small figure is all I have for that. The thiolytic cleavage involves addition of a CoA, breakage of this guy right here, and the attachment of the CoA at that place. So this leaves us with an acyl-CoA that's 2 shorter, and what we broke off was an acetyl-CoA. Now, acetyl-CoA can, of course, go to the citric acid cycle and be oxidized. So what have we seen so far? We've seen that one round of beta oxidation gave us one acetyl-CoA. It also gave us one FADH2, and it gave us one NADH. If we send those over to the citric acid cycle, that is the acetyl-CoA, we're going to get 3 more NADHs, we're going to get 1 more FADH2, and we're going to get 1 GTP. There's a lot of energy in a fatty acid. Okay? A lot of energy in a fatty acid. There's about 120 to 130, depending upon how you count them, ATPs that are produced per 16-carbon fatty acid. About 120 to 130, depending on how you count them, ATPs produced per a 16-carbon fatty acid. Per carbon, there's more energy in fatty acids than there is in glucose. Okay. There's the summary, blah, blah, blah. Oh, the last one, I forgot to tell you. So the last one, the enzyme in the last reaction is the other enzyme that's important. All right? The enzyme that catalyzes this reaction right here, this thiolysis reaction, it's called a thiolytic cleavage, is known as thiolase, Thiolase. Thiolase, and I have a dead battery. Thiolase catalyzes that break. Now, thiolase, we're going to see, is important later. Okay? Because what happens is thiolase can also run this reaction backwards and make something here. Okay? Not fatty acids, but it's involved in making ketone bodies that we'll be talking about later. All right? So thiolase is an enzyme to keep in your head because we will be talking about that later. Okay. Now we can get an idea of the rounds of oxidation. Here's palmitic acid. All right, long chain here. Break off an acetyl-CoA. Break off another acetyl-CoA. Break off another acetyl-CoA. If we start with 16, okay, how many breaks are we going to have before we finally get done? We're going to have 7 because the last one splits a 4-carbon one in 2. So we have 7 rounds of beta oxidation to break down a 16-carbon fatty acid into 8 acetyl-CoAs. Okay, so you see I'm breaking off 2 at a time there, and each one is pretty boring. Same sort of reactions going on over and over and over. Okay, now, this is all fine and dandy for saturated fatty acids. What you've seen is the simple scheme for saturated fatty acids. But our diet and our cells have things besides saturated fatty acids. They have unsaturated fatty acids. And unsaturated fatty acids make the picture a little bit more complicated. Why is it more complicated? Well, because they start with cis double bonds. And the enzymes that are set up for this oxidation are set up for trans double bonds. Okay? How are we going to handle that? Well, it turns out it's not as complicated, I think, as it is in words to describe. So let me show you. Here's a 16-carbon unsaturated form of palmitate known as palmitoyl, I can't even say it, CoA. Okay? It's got a double bond right here. If we go through several rounds of beta oxidation, which are shown here, we ultimately get a structure that looks like this guy. Here's carbon number 1, 2, 3, 4. Okay? Now, when we get to that position in this particular oxidation, we've got a cis bond between 3 and 4, and we want to have a trans bond between 2 and 3. If we have a trans bond between 2 and 3, this is an intermediate that's occurring in beta oxidation, so this will be okay to go and oxidize. So cells have an enzyme, and there's a name that probably you should know, big name, that's converting a cis 3,4 bond to a trans 2,3. Converting a cis 3,4 into a trans 2,3. Now this guy takes off and goes and gets oxidized by the regular beta oxidation route. Okay, everybody with me there? Out of this thing. This is not an energy-requiring process. This is simply an isomerization, and it doesn't require energy. I'm going to show you another one that does require energy in a minute. Okay, yes, Laurie.
But we get to feedback on that because you have to go through those first rounds
To get to this point. That's right. So the enzymes, if you think about it, are really only looking at this area. So anything that disturbs this area is going to be a problem for them. That area doesn't even appear until we have done several rounds of oxidation. Did I answer your question?
Well, no.
Okay. That would be the first time.
So I guess I'm piggyback and saying, does it take more energy because you have to go through those first rounds to get to where you transfer from the cyst to the bond, and then you go through the rest.
No, it doesn't take any more energy because the only additional step for this particular one is just this reaction right here. And since this reaction doesn't take additional energy, no, it doesn't take anything more to do that. So this would give exactly the same energy as, say, palmitoyl-CoA. Okay? All right. Now, there are other forms those unsaturated fatty acids can have, however. And if we look at this guy, it's a bit more complicated. There's always one of those, right? Oh. Okay. Well, let's look at this guy. Here's a guy that's got two unsaturated bonds. I'm going to keep it simple for you. Relax, okay? We're not going to get too upset at this, all right? Two cis bonds in this molecule, all right? Look where they are. So we go through several rounds of oxidation, and all of a sudden we've got 1, 2, 3, 4. Same thing as we saw before, right? Okay? Now, this is where your book actually has an error. I'm going to show you the error in just a second. But here's what happens. We get down to this guy right here, and that same enzyme as we saw before converts that 3,4 cis into a 2,3 trans. Okay? That guy can be oxidized by beta oxidation. So we're going to clip off 2 carbons. And we're going to get over here, and now what we've... I'm sorry, we're going to get over here, and now what we've got... Actually, let me back up. Yeah. Oh, wait. Now I messed up. Yes. Okay. So we're at this point, and we're starting the beta oxidation process, right? This guy, we've gone through this. We've 1, 2, 3, 4. Now I'm screwed up. Hold on. Yes. Okay. So I am right. Well, no, wait. I should have looked at this. All right, give me a second here. So 1, 2, 3, 4, 5. Yes, okay. So we do clip this guy off. We come down, and we are ready to now oxidize. We're ready to go the next round of oxidation in a beta oxidation. When we try to do that, we can go through the first step. So here's our saturated compound here. And we're going to put a double bond across here. We do that, and we make that trans double bond that we have here. Okay? Now, this trans double bond, okay, you would say, well, we can go ahead and we can add water across that and then go through the next oxidation. The enzyme that wants to put water on there doesn't like this double bond right here. It doesn't like the double bond right here. Okay? So these 2 double bonds in close proximity cause a problem for the enzyme that wants to add the water. What therefore has to happen is that these guys have to be rearranged. And the rearrangement is interesting. Here's where energy comes in. Okay? The energy that comes in, and this is where your book is wrong, okay, is in the form of NADPH. That's correct. And this enzyme name is also correct. And the fact that 2 double bonds are being converted into 1 double bond is also correct. What's incorrect is that's not a trans. That's actually a cis. So it's making not a trans 3,4, it's making a cis 3,4. Okay? So it's taking these 2 bonds and making a cis 3,4. Everybody with me? The reason that's important is because a cis 3,4 becomes a trans 2,3 with this same enzyme. This guy won't work on a trans 3,4. It only works on a cis 3,4. Now, I said I would keep it easy. Okay? Here's how you keep it easy. All you really need to know is what this enzyme does and what this enzyme does. This converts a trans 3,4 into a cis 2,3. This guy converts a cis-trans conjugated bonds into a single cis double bond 3,4. That's the bottom line of this figure. And that's really all you need to know. I'm not going to make you go draw this whole figure out. Don't worry about that. But I do think you need to know what those two enzymes are doing in general terms. Yes, Jenny?
What's the delta G0 prime?
What's the delta G0 prime of these enzymes? And I don't know. I couldn't tell you off the top of my head. That's a lot of material. So I thought we'd finish the week with a song. How's that? Okay. Okay, this is a song. It's to the tune of When Johnny Comes Marching Home. It's an old Irish song, okay? Everybody with me? Please sing along. The fatty acids carried by CoA are oxidized inside the mitochondria. They get to there, as you have seen, by hitching rides on carnitine. Then it goes away when acids get oxidized. Electrons move through membranes. Yes, it's true. It's true. They jump from complex 1 onto CoQ. The action can be quite intense when building proton gradients, and it's good for you when acids get oxidized. The protons pass through complex 5, you see. They do this to make lots of ATP. The mechanism you should know goes through the stages LTO, so there's energy when acids get oxidized. Okay, good enough. Yes?
Go ahead.
Yeah.
Is there a phosphatase that turns off the triglycerol lipase cycle?
There is, actually. So there's a phosphatase that turns that off, and it's stimulated by that insulin pathway. I haven't talked about it, but it's exactly like what we saw in the glycogen breakdown. Okay? Yes, sir.
A couple questions.
Okay.
First one, I asked you kind of a silly question by email, maybe Tuesday morning.
Okay.
Just kind of curious, this term gangliocyte, I mean, you actually have ganglion, which are collections of...
Yeah. I replied to your message.